It's the second one I've given today, actually.
Goodness. I think the slide is on now. Well, let's start now. A warm welcome to the Department of Chemical Physiology and Biochemistry and the 2021 Riker lecture. I'm really happy that this event actually happens despite COVID. Although we are extremely sorry that this is not an in-person event and that we have to do it all virtually this year. This is also the moment to thank all the people who had actually prepared this lecture for an in-person event, especially Amy, Ellie, Sierra from the foundation. A lot of people put a lot of work in, and now we had to funnel that all into the virtual presentation that you will be able to enjoy today. So I will start with giving an introduction to the Riker lecture and the endowment. This year marks the 100th birthday of Carmela Riker. the name giver of the Riker lecture. William and Camilla Riker got married in 1947. And after Bill had a strong career on the East Coast, in 1969, he was appointed chair of pharmacology here at OHSU, and they moved to the West Coast. And this position he held for 21 years. Camilla was very engaged in the US OHSU volunteering service and also an active member, and now mind these words, the Faculty Wives Club. Definitely something that we would have expected before the First World War, but no, it was still on in the 60s and 70s. Their tasks included the improvement of the patient facilities as well as student support, and the latter one was taken very seriously. Camilla was very dedicated to help others, but she also gave birth to 3 daughters, Gaynor, Ellie, and Vicky, and I think all 3 of them are actually listening in right now. After her untimely death in 1981, Dr. Riker initiated the Camilla Louise Riker Memorial Lecture Fund, which was fueled by the Riker family, but also by a large number of colleagues and organizations that all donated money to make this lecture series happen. The one was originally dedicated only to this invited memorial lecture to be given by a really outstanding scientist and educator once a year. But now that has been expanded to also support PhD students. Eventually, Dr. Riker married again, Dr. Lena Riker, physician at OHSU, who still lives in the area. And Dr. Riker himself then passed away in 2004. And subsequently, the Riker family decided to rename the endowment into the William and Camilla Riker Memorial Lecture. This is still a growing endowment, so people are continuing to make donations and put money into this endowment. We are very, very happy and very, very grateful for that. So since I never had the pleasure to meet the Rikers by myself, obviously I was an embryonic scientist when Dr. Riker retired, it is really difficult to do both of them justice. And therefore I collected a couple of statements from colleagues that did actually interact with the Rikers. And I'm now reading those statements to you. The first one is by Professor George Olson, who was a long-term member of this faculty here. And he states, Bill was a very kind, thoughtful, and experienced department chair who constantly had the interests of the students, faculty, and the School of Medicine at heart. He was able to make difficult decisions when required, but was always fair. His greatest professional interests included his research in synaptic transmission, teaching of pharmacology to medical students, scientific collaborations with clinical colleagues, and the development of drug metabolism research and a new discipline that then was called clinical pharmacology. Bill had a wonderful family, and his wife Camilla took junior faculty members and their families under her wing while raising her own 3 very active daughters and caring for her aging parents as well. So very busy life. Camilla was a wonderful hostess and accomplished cook, and she was very involved in what was then called the Wives Club. We smirked about that already, and that gave support for projects at OHSU. On a personal note, Bill loved New York City and Italian food, and George says, we have really missed them, but we were fortunate to know and to work with them. And Stephen Johnson, a former student of the Riker, states, as chair, Bill was a strong leader and administrator. During his tenure, he built the department So every facet of pharmacology was well represented by staff. He also loved the field and made sure we all knew the history, especially of cholinergic pharmacology. He was a strong mentor and taught a lot about editing and planning experiments. And he was a family man, so he clearly loved and supported his daughters. I think that gave you a little bit of an idea of what the Rikers were all about. Very group and family and socially oriented. And the last thing I will, before I introduce Peter Collins as a speaker, I will read a thank you note from the Riker daughters that fits in very well. So they thank the department for hosting this lecture and the 100th, especially on the 100th anniversary of Carmela's birth. Special thank to Peter Collins for working through several date changes to present the lecture today. The collaborative work and distinguished careers of our current and past speakers greatly honor our parents' memory and the community of shared knowledge and education that they valued so much. And we would like to share a quote from Dr. Sidney Spector, who presented the 1989 lecture, and I love this quote. I'm reminded of a Chinese maxim that if you plan for a year, you plant rice. If you plan for a decade, you plant trees. And if you plan for a millennium, you teach. And I think this speaks to the immense value science has in the quality of our lives. And thank you all for being here today and for learning, sharing, and teaching. So this was the statement by the Riker sisters. Thank you very, very much for that. And in this spirit, thank you all for joining us and picking up some real hardcore knowledge about how COVID vaccines and other therapies might work. And I will now start to introduce Peter Collins as the speaker. Before I do that, please, during or after the talk, email questions you have to events@ohsu.edu. And these questions will then be put into a WebEx, and I will read these questions to everybody, and Peter can hopefully answer them, I'm sure, very eloquently. So I know Peter from our participation in joint participation on the advisory board of a large Swiss research consortium. It was always great to work with Peter. Peter studied physics at UBC in Vancouver and received his PhD. Okay, I don't say the year. He then went on as a postdoc to the University of Oxford and changed from physics to biochemistry. In fact, he worked with Rex Richards there, who also changed from NMR and physics to soft physics, soft matter physics, which were membranes. And, you know, Peter stayed with that. He also then did a very short postdoc stint in Utrecht, which at that time was really the epicenter of lipid and membrane biology and biochemistry in the Netherlands. Then he moved back to Canada. He was appointed assistant professor at UBC and is a full professor since 1985. Peter is particularly interested in lipids in biological membranes and the development of nanomedicines using lipid nanoparticles. And I assume we will hear a lot about this topic today. Peter has published more than 350 papers, holds over 50 patents, probably at 60 by now, and his H index is at about 120. That is truly remarkable. But the most remarkable thing, I think, is that Peter co-founded and directed at least 13 startup companies and 2 Canadian centers of excellence. He is probably responsible for a very significant portion of the biotech industry in the Vancouver area. And one of the lipid nanoparticles that were developed by Acutus is currently in clinical phase 3 with BioNTech and Pfizer. And you can already imagine that this will penetrate into future COVID and vaccine developments. Thank you, Peter, so much for presenting this year's Riker lecture on a highly relevant topic. The title is The Science Behind Your COVID Shot, Lipid Nanoparticles, Gene Therapy, and COVID-19 Vaccines. I'm sure Dr. Riker would have approved of such an important and current topic. Peter, the stage is yours.
Well, thanks very much, Karsten. Just want to check first of all whether people can see my screen.
Not yet. You'll have to jump on and share, Dr. Collis.
I'm going to have to do something else here then. I just sure that. How about now? So is that working?
Yes.
There we go.
Thank
Okay,
you.
great. So thanks very much, Karsten. And I really regret not being there in person. It is too bad. We did try a couple of dates, and they just didn't work. I also want to say to the Riker family what an honor this is. I looked at some of the previous speakers and realized that this is a pretty significant event. And for those in the family that are listening, I really am very much honored and touched by this, by this named lecture, or being able to give this named lecture. Now, what I'm going to talk about today is, the title is "The Science Behind Your COVID Shot," but we cover a lot of ground here. I am also cognizant of the fact that I think about 20 to 30% of the audience are non-scientists. So I've tried to put some things in terms that may be maybe more able to understand. So it has resulted in an expansion of the number of slides I've got. So if you notice, I'm moving quickly at some points. That's simply so I want to be able to observe the time frame that we have. I have a couple of conflicts of interest, precision nanosystems that were germane to this talk. Precision Nanosystems, where I'm a founder, and Acuitas Therapeutics, where I'm also, which I'm also a founder. But I'll be indicating some of these companies as we move through. Okay, if you've been following the news about the vaccines, the mRNA vaccines in particular, you'll see frequent mention of tiny bubbles of fat. This is a headline from Bloomberg. The vaccine revolution is coming inside tiny bubbles of fat. And so that's I'm just going to put this pointer on here. This is this little globule here, which is an integral part of the vaccine. And so this sort of description is actually fairly rampant in the popular press. And so what I'm going to be talking to you about today is okay, well, what are these tiny bubbles of fat anyway? Well, first of all, they are pretty tiny, 100 nanometers in diameter, which is 100 times more smaller than a cell. And you have roughly, well, a few hundred trillion cells in your body. That's 100,000 billion cells. So they're pretty small, about 10 microns or so. Just to give you a sense of proportion here, if you have a pen or a pencil and you draw a line on a piece of paper, you can fit roughly 10 cells in the thickness of that line. And so at 100 times smaller than a cell, you can get 1,000 of these nanoparticles into the thickness of a line on a piece of paper. So they're pretty small, but they're not really bubbles of fat. This is, at least it's not the fat you normally think of. They're rather sophisticated particles that are made of what we term membrane lipids, and they're different from the lipids that you normally associate with, say, a fatty meal or whatever. So I'm telling a story here that started over 40 years ago. I'll go through a little bit about how I got involved with membrane lipids and their properties, and then move to, well, how can we use these kinds of systems, these lipids, to encapsulate nucleic acids such as messenger RNA, and also to encapsulate proteins such as DNA. And then finally, and how can we use these systems to deliver mRNA, and how we got into the vaccine area. What I'm going to emphasize here is essentially the nonlinearity of science. You end up uh, uh, doing things that are very hard to predict at the outset of one's career. So how did I get involved with membranes and lipids? As Karsten has indicated, a PhD in physics and a postdoc in biochemistry in Oxford, in Utrecht, subsequently in Utrecht. I've been a professor here in the biochemistry department at UBC, biochemistry and molecular biology, since about 1978. Been a few changes over the 50 years, nearly 50 years that I've been doing research. So PhD was done using magnetic resonance, and you're familiar with magnetic resonance from magnetic resonance imaging, but I was using it in a different form to investigate the properties of semiconductors at 4.2 degrees Kelvin. You can think of this as actually looking at the properties of transistors on the moon. 4.2 degrees Kelvin is just above absolute zero, so it's pretty darn cold. um, I realized I didn't want to keep on doing this kind of thing for the rest of my life. I found it very difficult to explain what I was doing to girlfriends, and so that was one one worry. The other was that some of the problems outside physics just seemed way more intrinsically interesting. So I did a postdoc using NMR, magnetic resonance, to study, and I was studying lipid polymorphism then, which is the ability of membrane lipids to adopt really quite different structures. And then while I was on faculty at UBC, continued with these studies of lipid polymorphism, and then another topic called lipid asymmetry. All of these have relevance in the end to the COVID-19 vaccine, so I'll be tying it together a bit towards the end. Then we started to deliver anti-cancer drugs, and then nucleic acids, and then that changed into something that ended up with the COVID-19 vaccine. All I can say, given this career path, is that science is a very non-linear endeavor. I've had a lot of help. As Karsten said, I started quite a few. I think Karsten said 13. It's about 12. I'll explain how we got started and why it is that this has been been very helpful in many ways. So forward, germane to this talk, first company we started was Lipex Biomembranes. That was in 1985. Inex Pharmaceuticals in 1992, Protiva Biotherapeutics 2001, and then Acuitas Therapeutics in 2009. So a large part of the work that I'll describe here was conducted at these companies. I was intimately involved in the direction or the science that was being done in those companies over this time. So let's start at the beginning. I got as I mentioned, my PhD in solid state physics, but it's after graduating, I decided that I had to do something different. As I mentioned, it seemed that the most interesting problems were outside the field of physics. I got vaguely interested in biological membranes, even though I didn't know much about them. And by good luck, I was awarded a Medical Research Council fellowship, even though I was a physicist, to go to the biochemistry department in Oxford in 1973. Now, what do you learn as a physicist? You learn quite a few things, but I just indicated three here. One of the first things you learn is that you learn a technique, but you don't just learn technique, you learn it in real depth. This is my last physics paper, which was published in 1976 in the Journal of Magnetic Resonance, a very theoretical paper consisting of about 96 equations that was at least allowed me to say I was leaving the field with honor. But it does point out that you do learn things in real depth, and that was very handy in my subsequent work. The other thing you learn as a physicist is that when you need an instrument that isn't available, you build it. So this is me in Oxford in 1975. And so I arrived there, and there was the makings of an NMR machine. There was a magnet. That's this entity here, but there was nothing else. We had to build all the rest of this spectrometer. And so me and a graduate student, we put this thing together. So you get used to building stuff as a physicist, and that certainly turned out to be very handy in this circumstance. The third thing is you go after basic problems, and so this is a model of a biological membrane, as I mentioned. This surrounds every cell in your body, as well as being made use of inside the cell for other functions. So there's biological membranes there's some basic problems here. They contain hundreds of different species of lipids, and why are they all there? So it's a basic problem. It still is a basic problem. And the membranes, biological membranes, have different lipids on one side compared to the other. How's that generated, maintained, and what are the functional consequences? So this is the basic work that I started off on that fed right in to the vaccine, as I'll make apparent. So we have this from a cartoon point of view, the lipid bilayer, which we can do in cartoon form, indicated here. So membrane lipids have a very particular quality. They're amphipathic. They have one end that likes to be in the close to water or in water, and the other end that's hydrophobic and likes to be sequestered away from water. And so that's why they will preferentially adopt this bilayer structure. So so that's what we're going to be talking about, is really these types of membrane lipids and what their properties are. And so one of the structures that lipids in membranes adopt is this, aside from this bilayer structure, and you can certainly see the function of the lipid bilayer. It separates the inside from the outside. But another structure that, if you take a lot of lipids out of membranes and put them in water, they adopt this hexagonal H2 phase, which consists of a tube of water surrounded by these the polar head groups with the acyl chains, the hydrophobic portion sequestered away. So what are they doing there, and why do they adopt these structures? And similarly, with lipid asymmetry, we started to ask the question, well, interesting, this lipid distribution, can this be generated in response to, there's always ion gradients across biological membranes, the sodium, potassium, protons. So this is basically the acidity of one side compared to the other. And so what are the consequences of these asymmetric transbilayer distributions? And what we did while in Oxford and subsequently was to use phosphorus NMR. That's all these lipids are mainly phospholipids. They have a phosphorus atom in the head group, and there's I'm not going to go into the theory behind this, but if you put these lipids in water, they will adopt various structures, and you get very characteristic phosphorus NMR signals resulting from that, from the lipids either in a bilayer or being in this hexagonal 2 phase, or if they're in other structures that are also available to them. And so we could use phosphorus NMR to study lipid polymorphism. And we found we could explain the the polymorphic phase preferences of lipids using a so-called shape hypothesis. It's a really simple idea, but if lipids have different shapes, different areas in the head group versus the acyl chain versus the hydrophobic part, then they will adopt, say, micellar phase. If they're cylindrical, a bilayer phase. If they're cone-shaped, then this hexagonal phase. This was done with Ben de Krouff, who was a postdoc with me in Oxford, and then subsequently came and worked for a year here in Vancouver. And we made a great deal of progress in this area. Now, in order to investigate lipid asymmetry, I'll be coming back to the polymorphism subsequently. It's very directly related to the vaccine application. Now let's go on to lipid asymmetry. Now, what we needed here, we needed a bilayer system in which we could induce lipid asymmetry. So first of all, we had to be able to make these unilamellar systems, and there were no really good techniques for doing that at this time. So we devised this process where we forced much larger lipid constructs through a polycarbonate filter that had 100 nanometer pore size, and out the other end came these unilamellar vesicles. So it was really it's another example of when you need an apparatus that isn't available, then have a go at building it. And so we were able to generate these kinds of systems in a reasonably high throughput and short time frame. Now, it turned out that extruder was a pretty popular device. I started to get all sorts of people saying that they wanted to buy one. So we started a company to sell it. This was the start of our our company endeavors. We knew this was the board of directors of WebEx Biomembranes, which was what we called the company. So it was me and 4 postdocs that started it. We knew that companies had to have mission statements, and so we decided our mission statement was going to be free beer every Friday and parties whenever possible. Nobody in my lab has paid for beer on Friday since. So we found we enjoyed starting companies. Now, we use these systems to show that membrane fusion, say when 2 things fuse together, really depended on the presence of these non-bilayer lipids. And we could also use these systems to generate lipid asymmetry. Now we come on to something you're going to hear a lot about later on. This is ionizable cationic lipid. This is a lipid that can exist either in a neutral form. So this is a tertiary amine in the head group. So it can be either deprotonated and net neutral, or it can be protonated and then positively charged. So we synthesize this lipid. It has the property, if you have a pH gradient across this lipid bilayer, say an inside is pH 4, that's about the acidity of a lemon, and outside physiological pH and pH in your body. We found we could drive lipid asymmetry. The ionizable lipid will preferentially distribute to the side of the membrane that experiences the low pH. And so we could drive all sorts of things doing this. If we moved lipids from one side to the other, say from the inside to the outside, we could drive shape changes from the stomatocytes, as it's termed, sort of shape, to shapes that are more compatible with having more lipid on the outside than on the inside. And so these long elongated structures. Now, I'm pretty sure that biological membranes actually use this kind of phenomenon to advantage, but at this point, we got very distracted. We got distracted because what we found was that we could load drugs into these systems using very similar approaches. This is just indicating how we measure the pH. It's using methylamine, the small molecule here, which can exist in a neutral form and a positively charged form. The neutral form is highly membrane permeable, and so it gets protonated on the inside in the low pH environment. Can't get back out because charged things don't permeate through membranes very easily. Equilibrium, you could get 1,000 times high, say a pH gradient of 3 units. That really means that we could get it at equilibrium 1,000 times higher concentration of methylamine on the inside and on the outside. That's how we measure the pH. But we made an observation here that many common drugs are weak bases. So that's just a few cancer drugs indicated here. You can see that, say, doxorubicin, which is a common anti-cancer drug, is a weak base, or it has this tertiary methylamine, or aminofunction. So we found we could load drugs inside these systems to such high levels that they would pass their solubility product and they would precipitate out into nanocrystals on the inside of these lipid nanoparticles or liposomal systems. This is a bar indicating 100 nanometers in diameter. So you can see that this is a very efficient way of loading these very small systems with cancer drugs. So we started another company. This was Ionex Pharmaceuticals, with the objective of trying to get cancer drugs much more specifically to where you need them, as opposed to going all the way through your body. Now, I'm not going to go into any detail here, but we did get 3 drugs approved by the FDA and the European Medical Agency to do just that, particularly with doxorubicin and vincristine. But the story here actually turned us onto a completely different path. So we started Ionex Pharmaceuticals, as I mentioned, and then the CEO, Jim Miller, came to me one day in the middle 90s and said, we can't raise money for the company putting old drugs into liposomes, as these nanoparticles are known. We need to be doing gene therapy, which is in vogue at the time and has much more sex appeal. So this started us off on a whole different path. So what is gene therapy? There's a Wikipedia definition, pretty general. It's the therapeutic delivery of nucleic acids into a patient's cell as a drug to treat disease. But the key word is delivery. If you don't, if you don't have a delivery system for these nucleic acid-based drugs, they can't get in. They tend to be rapidly degraded in biological fluids. They don't preferentially accumulate where you want them to accumulate, and they can't penetrate into target cells even if they get there. So you're really hooped without a delivery system. Just to remind you about the central dogmas as termed in molecular biology, which is DNA goes to RNA, to messenger RNA, and then this is in the nucleus and this is in the cytoplasm. This is transcribed, the ribosomes, and then we have a protein that then does the actual work inside the cell. And so there's a lot we can do without going into the nucleus, and particularly with RNA-based RNA-based approaches. So what we did was we focused primarily on these RNA-based opportunities. A molecule is called short interfering RNA to inhibit production of proteins, pathogenic proteins if you want a therapeutic. And then the messenger RNA, if you can get that into the cytoplasm, then it can be transcribed to make a particular protein. But the trick is we got to get these things inside the cells. I should say that the potential here is huge. One can conceptually at least devise ways in which this could treat pretty much all diseases, certainly most diseases that people suffer from. So now we get to the core of the core of the matter. How do we develop these lipid nanoparticles to deliver mRNA into cells in vivo? So this has three parts. First of all, how do we encapsulate these nucleic acids? Still ongoing. Second, what was the first demonstration that we could actually do this? And this was done for short interfering RNA, which silences a particular protein. And then moving on to the vaccine application, which of course uses messenger RNA as opposed to siRNA. So messenger RNA, we're now getting gene expression. So our initial aim back in '95 was to say, okay, well, let's try and develop a delivery system to enable siRNA to silence a gene in the liver. So going in intravenously and then having the circulate around for a little while, getting to the liver, then getting out of the circulation to be able to make contact with the liver cells, with the hepatocytes in the liver, and then getting inside the hepatocytes and delivering the contents to the inside of the cell. Now you can tell this is not easy to do. Nature has devised very intricate ways of preventing genetic information from getting into the cells of a host. The immune system is particularly triggered or designed with this in mind. But we had a first problem. How do we encapsulate lipid nanoparticles into, how do we encapsulate these nucleic acid polymers into lipid nanoparticles? We really require positively charged lipids to interact with a negative charge, high negative charge on the RNA or DNA. There's just one major problem, and that is there are no cationic lipids in nature. They're either net neutral or negatively charged. Cationic lipids are highly toxic molecules. And we knew from our experience with the cancer drugs that toxicity will always kill you in the end if you don't pay attention to that right from the outset. So what we did was we tried DODAP, which was the lipid we synthesized to investigate lipid asymmetry. It has this property that it's not charged, it's at pH 7, 7.4, but if we go down to pH 4, then it's positively charged. And so we asked the question, can we encapsulate the nucleic acid polymer into lipid nanoparticles? essentially by going down to pH 4, where the cation lipid is going to be positively charged, and then taking advantage of the association between the cationic lipid and the S, in this case, S small interfering RNA. So we devised a way of doing this. This is again another example of building an apparatus where we mix together all the lipids dissolved in ethanol, and then mix them very rapidly with with an aqueous solution with water containing our nucleic acid that we wanted to encapsulate. Now, if we do this fast enough, the first thing to fall out of solution as you raise the polarity is the oligonucleotide surrounded by the cationic lipid. Now, if you do it very slowly, of course, these are all going to aggregate, and you end up with a great big blob of material. If you do it fast enough, and then you have other lipids that are more surface lipids, like these polyethylene glycol lipids. They will precipitate out, and then they will coat these systems to achieve what we term as the limit size, which is the smallest size that's compatible with all the molecular components that you have in there. So you then remove the ethanol and raise the pH, and we found that it did retains the contents inside these lipid nanoparticles. So these were new systems that they have a different hydrophobic core, ideally encapsulated suited to encapsulation of the negatively charged macromolecules. Encapsulation efficiency is approaching 100%. We could dial the size by changing the ratio of surface lipid to core lipid. It was relatively scalable and reproducible. So first part, okay, we could do this, and we had something that was reasonably non-toxic. Now, is this of any use? And that's what I'll talk about now with regard to what's termed the Onpattro story that took place between about 2005 to 2012. So we're asking the question, if we trapped small interfering RNA in here, can we silence genes in hepatocytes following an intravenous injection? Now, how do we get these lipid nanoparticles into cells? What we use here is endocytosis. This is the way that cells eat. They will take stuff in using these small endosomal vesicles, transport it through to a lysosome where everything gets chewed up, and then the cell can use the constituents of whatever amino acids, etc., that might result from that. So this process is quite it's a very precise, well-regulated process, but it depends on, as you go down through this progression, the pH on the inside of the endosome is reduced to, first of all, to somewhere in the region of 6, between 5.5 and 6.3, and then subsequently below 5, 4.5, or something in that region. And so we had to design what we wanted to do was design our systems so it will escape from the endosomes before, of course, they go on to the lysosomes where they're all going to get chewed up. And so we had to design these systems so that they could maximize endosomal release. So this was where we got back to lipid polymorphism. Because what we did about 20 years ago was say, well, ask the question, why is it that these cationic lipids can, at least in vitro, the permanently positively charged ones, how is it that they can actually get these big molecules into cells? And what we found was that you have a cationic lipid, here is an example of DODAC, and added to one of the negatively charged lipids that you find in biological membranes that flips it over to this non-bilayer phase. And so this is just, you can think of it in terms of the shapes of the molecule, makes it more cone shape. So they both support a bilayer in isolation, but together they'll adopt this structure. And so, of course, this would be very membrane disruptive. And so we designed our ionizable lipids, and so that they would maximize the potential for this non-bilayer structure at a certain point as the pH was lowered in the endosome. So we had to design our systems to have the maximum cone shape, as I indicated, and also so that they become positively charged at some optimal pH. The point at which they become positively charged depends on what's termed the pKa the apparent PK of these molecules. So we collaborated with a company in Boston called Alnylam Pharmaceuticals to develop these systems. We set up teams here in Vancouver and in Boston. I worked for 7 years. We synthesized over 300 different lipids, and varying the head group and varying the pKa and varying the shape properties. And we ended up with systems that had a therapeutic index for silencing genes in the liver, a therapeutic index of well over 1,000. So we used what's termed well, at least what we were using was a factor 7 model. So we had siRNA to silence a gene that's made in the or protein that's made in the hepatocytes. Factor 7 is one of the clotting proteins. So we could inject these lipid nanoparticles containing siRNA using our rapid mixing process. They were made using that process. So we inject them intravenously. They go to the liver and inhibit the production of factor 7. And then 24 hours later, we can terminate the mice and assay the plasma for factor 7 and see how active our system was. And so we found that these were highly sensitive to the different ionizable lipids. And we ended up with this lipid, as I mentioned, that had this very high potency with relatively low toxicity. It's called MC3. So here's where we started. We started in order to get 50% gene silencing, we had to give as much as 10 milligrams. We were very close to killing the animals with that dose. And over the course of these 7 years, we got to this 5 micrograms of siRNA to get the same gene silencing without increasing. If anything, we decreased the toxicity of these entities. So what we can do now is by extension, we're silencing factor 7 using this lipid nanoparticle system. This is one of the great things when you're working with gene therapies. By extension, we could silence any gene. We just had to change this siRNA so that it would associate with messenger RNA for a different protein. And so this is where the clinicians then said, we were asking the question, what gene should be silenced and will this work clinically? And so they chose to develop a therapeutic to treat a disorder called transthyretin-induced amyloidosis, which is, of course, a disease you've never heard of, but it is actually a pretty nasty disease. So transthyretin is a protein that's made in the liver, made in the hepatocytes, and it is a retinol binding protein, as it's termed. It has this property that is pretty large protein, and mutations in the gene can lead to a formation of these fibrils, which really will deposit anywhere in your body, but it has particularly nasty ramifications if it deposits in nervous tissue or in heart. So there's neuropathies and cardiotoxicities that result. There's no effective therapy that's usually fatal within 5 years of diagnosis. So this just indicates the progression of this disorder. You can see it's a very nasty progression. It's also a wasting disease, particularly nasty when you consider that individuals afflicted with early symptoms will very likely have a relative who's had a similar condition, and they'll have seen the progression of the disorder. So the hypothesis here was very simple. Let's use our lipid nanoparticle containing the small interfering RNA, in this case now to silence transthyretin. We reduce the transthyretin that's being produced, and so therefore we should see stabilization, maybe even if we're very lucky, recovery from the cardiotoxicity and the neurotoxicity. So this is a feature actually of gene therapies in general if you get them to work. You can think in a very basic way about how you might treat disease such as the transthyretin, such as this amyloidosis. Phase 1 results from healthy volunteers, we showed that if we gave a dose, or the clinicians showed that if we gave a dose of 0.15 or 0.3 mg per kg, you get very rapid silencing of the transthyretin in the blood that's apparent in the blood that lasts for about 2 or 3 weeks. And so they selected a dose of about 0.3 mg per kg every three, or not about, exactly 0.3 mg per kg body weight every 3 weeks for the subsequent trials. And so this, moving on to the definitive trial, which is the phase 3 study. This is a double-blind study that was done with some patients getting placebo. Of the 225 patients, 77 got the placebo, 148 got the drug that's silencing the transthyretin gene. And we're looking at the primary endpoint over the 18-month course of this clinical trial was the so-called neural impairment score, which is a measure of the neuropathy of the nerve toxicity we might be seeing from this disorder. There's various other secondary endpoints that were also measured. And so what was what was found here was that this was one of the most remarkable phased results from a clinical trial that I've ever been, or at least I've ever seen. We hit the primary endpoint and all secondary endpoints with p-values, confidence values that are beyond belief. I mean, this drug obviously works. For those of you that are more physical minds, this is 1 over Avogadro's number. This is pretty convincing. This is in terms of the neural impairment score. And the quality of life, this is self-reported quality of life, muscle strength, better than placebo, the disability, there's a gait speed, nutritional status, etc. All of those conditions were ameliorated. So you're actually getting making people better, even though they're suffering from a hereditary disorder. So this drug is a stabilizing and possibly curative therapy for previously fatal disease. As you can imagine, we were pretty ecstatic about these results, and this was approved by the FDA in 2018. It was the first FDA approval of an siRNA-based drug. So as I just indicated, a pretty big deal that dramatically demonstrates the power of gene therapies. Okay, so now let's go on to the final part, which deals with where we end up with the vaccine application. And so this took place at Acuitas primarily from about 2012 to 2020. So we're asking the question now, does the lipid nanoparticle technology that we developed for siRNA, where we're getting gene silencing in the liver, does that enable mRNA-based gene therapies? And at Acuitas, we wanted to find that out. So it's a very similar process to what we were doing with the messenger RNA, where we were, but now we're encapsulating for the siRNA. Now we're encapsulating mRNA into these lipid nanoparticles, injecting intravenously. And then we're asking the question, well, if we can get the siRNA into the cytoplasm of the cell, maybe we can get mRNA in there and actually then have that protein being synthesized. And so what we found was that that this worked. This is at least initially with the MC3 lipid that was the optimized lipid for siRNA. This is using luciferase and looking at live imaging following 4 hours following an intravenous administration. And you can see there's a dose dependence here over the for these animals. We can visualize the increase in luminescence in the liver, and it's proportional to the dose that we give. And so that's where Acuitas took this and then said, okay, well, can we improve on that? And so this is starting with looking at the luciferase expression that we began with, and this is about 2012 or 2013. And now they've got systems that are some 30 or 40-fold improved over that. So this improvement of the ionizable lipid has gone on to give us really it's one of these. It's a gift that keeps on giving. That's for sure. These ionizable lipids have really solved a lot of the problems. Now, in 2014, we were approached by Drew Weissman, who's an immunologist at the University of Pennsylvania. And he'd been following our work, first of all with siRNA and then subsequently with mRNA. And he wanted to do was to say, okay, well, this is all very interesting. You're getting gene expression in the liver following an IV injection. But what I would like to know is can we get gene expression if we go in, if we have a mRNA that's coding for a protein that's associated with a virus, and if we inject that intramuscularly, can we get a reasonable immune response? He'd been working for some time with Catalin Carrico, and what they'd shown is by modifying mRNA, they could reduce immune activation and increase gene expression. But this was in vitro. So they said, we want to be able to do this in vivo. So how do we get this mRNA into muscle and immune cells? So this is an example of serendipity that can happen when you're working in one direction and then suddenly you find yourself moving in a very different direction. So Drew needed a delivery system to enable these mRNA vaccines. First of all, to transfect muscle cells and then antigen-presenting cells that were in that region, and perhaps they were drained to the local lymph node and transfect cells there to produce a robust immune response. The MHC1, MHC2 fast responses. What happened here was quite remarkable. That so Acuitas was sending material to Drew. He was then in pretty good contact with a number of labs that had very good animal models in a variety of areas. He would send the material to them, and then about 2 weeks, 3 weeks later, whatever the time frame was, he'd get these phone calls or emails saying, "Well, gee, why did you send this? This stuff really works." And so one example of that was for Zika virus. And so what was done here is to develop messenger RNA that coded for the Zika virus pre-membrane and envelope glycoprotein, as it's termed, package that into the lipid nanoparticle. And so by doing that, hope to raise an immune response to the Zika virus. So this is work published in Nature in 2017. And what was observed was total protection against the Zika virus from a challenge. And so here what was done was injecting intradermally the lipid nanoparticle containing the mRNA coding for this a protein. And then subsequently injecting IV the Zika virus itself at a pretty high dose. So as you can see here, if the mRNA was coding for luciferase, you didn't see any protection. You see some pretty high viral loads, but total protection for the animals that were injected with the mRNA that codes for the viral protein, both at 2 weeks and at 20 weeks. Again, total protection. Acuitas was also working with a company in Germany called CureVac, and in January 2020, they actually managed to get a clinical trial finished, and they showed that for a rabies vaccine, so in this case, the lipid nanoparticle contained mRNA for a glycoprotein that's on the outside of the rabies virus. And the clinical trial results show that at a very low dose of 1 microgram, there was very high levels of neutralizing antibodies in these patients. And so this looked pretty good. But Acuitas was also working with BioNTech at this point to develop influenza vaccines. And BioNTech, in turn, was working with Pfizer. And of course, all attention switched to COVID-19 in January, February timeframe last year, and and so what was decided to be done, and this was a real gamble in many ways on Pfizer and the BioNTech's part, but they switched all efforts to making a vaccine where they were incorporating messenger RNA coding for the SARS-CoV-2 spike glycoprotein. This is the glycoprotein that's associated with the COVID-19 disease. So this was, I think everybody knows what happened after that. The results of this trial were announced in November of 2020. That the vaccine, this is the Pfizer BioNTech vaccine, was 95% effective against COVID-19. This was overall ages and ethnic groups, gender, etc., reasonably well tolerated, no serious safety concerns, and that at that point they expected to produce 50 million doses in 2020 and 1.3 billion doses by the end of 2021. I think they're up to 3 billion doses that are prepared this year, and they are producing 50 million doses a week at the moment. So this has been approved, as we're all aware, in a large variety of jurisdictions, and is playing a pretty big role in ending the COVID-19 pandemic. So this is real world efficacy in Israel. So starting vaccinations, and you can see the effects on the new COVID cases over that time frame. Now we are seeing that this is starting to go up again, so booster shots are very likely to be necessary. But there's little doubt this is playing a major role in treating the pandemic. So this is the journey that we've been on in terms of the applications for the RNA-based drugs, both siRNA and subsequently mRNA, and it's really been quite a journey. One thing to note is that this is really the tip of the iceberg. We're going to see vaccines coming along for not just COVID-19, but HIV, Zika, probably a universal flu vaccine. The approaches here that are becoming apparent for treating cancer, heart disease, Alzheimer's, various chronic diseases are really quite tangible. And of course, for rare diseases, inherited diseases, where you might be suffering from a particular protein that's not being made, when say a child is born with an absence of a clotting protein, whatever, you can get the liver to make that, and you can have that designed and made in a time on the order of a couple of months. So it's a very exciting time. Just on a word on that, so a very rapid and precise way in which we can develop these gene therapies. Once we identify the protein we want to silence or express or edit, and we can use siRNA to silence it or mRNA to express it or to edit it, we can encapsulate that in a lipid nanoparticle. And a new medicine can be available in a time on the order of weeks. I mean, just think about that, a very highly targeted medicine that when we're used to, say, small molecule drugs that might take 10 to 15 years to develop. So just in closing, I just want to say all of this arose from, as I was trying to point out, very basic research many years ago, asking the questions, why are there so many lipids in membranes and what are they doing, lipid polymorphism, and the questions on lipid asymmetry. How can we induce lipid asymmetry and what are the consequences of that? So the results that I've shown in terms of therapeutics, we wouldn't have got there if we hadn't been asking these completely unrelated questions many years ago. So it's resulted in 5 drugs that have been approved in the U.S. and Europe and Canada, and of course is the work reflects the efforts of literally hundreds, probably closer to thousands of people. A few people I want to really acknowledge, because the same people that I showed you pictures of at the beginning, say when we started WebEx biomembranes, they're still working with me today. So 40 years later, it has been a large part of the reason for our success is that we've built up this very long-term center of excellence in the lipid technology. Steve Ansel, Ying Tam, Bar Mui, all of these people I've worked for well over 20 years, and the list goes on and on. So the Ian McLaughlin, James Hayes at Arbutus played a big role. The team at Alnylam, Mark Tracy, Akina Kink, and Martin Mayer, Manu Manoharan, we broke the problem in many ways. We were working with them on the small interfering RNA, along with Marcus Cipollini, who played a pivotal role in that. Of course, my own group at UBC, and then Drew Weissman, who, in terms of the vaccines, well, these are serendipitous events you can't predict, but it certainly has worked to our advantage. So I'll close there and take any questions people may have.
That was fascinating. Thank you so much. I'm really amazed to see how very basic science makes it really into the patient and has such a bright future ahead. And it might also explain to people that these vaccines that have been stuck into so many people, billions of people by now, did not come down on us within a year or so, as some people might have thought. There was decades of work done before that made this huge success, almost miraculous success in getting that rapid treatment for something that threatening into the patient. I do think this is a great example that science has good chances to really make a huge difference in the life of a large portion of our population. Thank you, Peter, for making that so clear and give us this beautiful example, and congratulations on all the fantastic results you and all your co-workers provided for us.
Thanks very much, Karsten. No, it has been quite it has been certainly amazed me, you know, how, as you say, very basic science gives you tools that you can then say, oh, I can use that in a completely different way. So it has been quite a, yeah, we're still working in some ways on lipid asymmetry and lipid polymorphism. I mean, they're fascinating topics, particularly as it relates to the behavior in the endosome. There's a lot still to do there.
So we will have to go to this after lecture question answer session now, and I'm gonna see you and everybody else hopefully there in about 10 seconds or something like that.
Okay.
And just real quickly, if you are going to be joining that, there is a link in the description on YouTube that you're watching. So just kind of scroll down, and in that description, there's a clickable link for you to be able to join for additional questions for Dr. Coles. Thank you.